Good afternoon, everybody. I'm Krish Roy. I'm professor of biomedical engineering at Georgia Tech, and I wanted to welcome you to this afternoon session on scaling manufacturing of cell therapies, current capabilities, key strategies for moving forward. We have a wonderful panel today, but before that, I wanted to thank the Regenerative Medicine Foundation, the Alliance for Cell Therapy Now, the Cura Foundation, and the Cord Blood Association for making this day possible. It's been a fantastic day with great discussions and talks, and we are excited to join that conversation from a more manufacturing and quality control aspect. So we have three panelists today. John McMannis, Executive Vice President of Manufacturing at Mesoblast, Lisa McClain-Moss, Senior Director of Manufacturing at Longeveron, and John Rowley, Founder and Chief Product Officer at RoosterBio. So all from industry, and I think what you will hear today is where the cell therapy industry, or the sort of the non-T cell therapy industry is, and where things are moving forward, how we are thinking about scale and quality and standards, and so on and so forth. From my own perspective, as you probably heard from the noon session with the FDA, the word manufacturing came up in almost every other sentence, if not almost in every sentence. And manufacturing and CMC and quality control remains, one of the most critical aspects of cell therapies, especially when we think about safety, efficacy, and scale. So we are going to discuss many aspects of that today in the time that we have. But what I would like to do is start by simply asking each of the panelists to introduce themselves a little bit, talk about their organizations, the products they are manufacturing, what maybe make a comment about current scale that they're at or they can be, and sort of broad comments about their general approach when we think about CMC and manufacturing in the cell therapy space. And although most of the companies here today work on a specific cell therapy areas, I will ask them throughout the conversation to put a broader hat about their knowledge about the broad cell therapy space, and their comments kind of relevant to that space. So maybe I'll go with John Rowley first from Rooster. John?
Sure. Hi. Thanks for having me, Krish, and the organizers. RoosterBio is not a therapeutic company, so we're a little different than the other two panelists. But we do focus our company on solving the challenges of MSC, large scale manufacturing. What we are is we've taken the traditional cell and media business model that many people have seen in the research field, and we've focused that model on a unique customer segment, which is the regenerative medicine product developer. And it's these companies that are wanting to develop regenerative medicine products that incorporate an MSC component, whether it's the main component or even a small subcomponent. And what we've done is and are still continuously trying to do is really standardize and industrialize the supply chain for these products that want to incorporate MSCs. We provide, like I said, vials of cells and bottles of media like dozens of other companies out there. But ours are CGMP grade, and they're built for scalability with a manufacturing process associated with it. So essentially, we sell our vials of cells. They're actually CGMP released working cell banks. Now we have research use only versions of these for that are lower cost and for product development and manufacturing process development, and then we have a bioprocess media. Which works as in both flasks as well as bioreactors. And so we've really tackled that transition, from going from two-dimensional flask-based culture into scalable 3D bioreactor culture. And we've been lucky to receive some of the early MTech funding, which is the Medical Technology Enterprise Consortium for specifically for bioreactor-based manufacturing processes, and they helped fund the development of a 50-liter completely xeno-free process for MSCs. So taking our starting out with a working cell bank MSC with a low population doubling, well below 10, and then getting it to a standardized population doubling around say 16 to 18, within a large scale bioreactor. With the goal of getting those cell densities well north of 500,000 cells per mil. And I think, going from scale, while our own scale, since we manufacture the working cell bank, is in the low billions, or maybe the single digit billions, these working cell banks, when they're expanded, our client processes need to accommodate anywhere from 5 billion at the small scale to, out of a 50-liter bioreactor, you can definitely get at least 30 billion cells, and that's before really pushing the densities. And I would say we both provide working cell banks from bone marrow as well as umbilical cord derived MSCs, and umbilical cord derived MSCs, because they have actually a younger. They're very different tissues, and some of the characteristics of these different tissues are very different, and so they're going to be good for different indications. I'm highly confident of, but umbilical cord cells actually have some manufacturing benefits where they grow a little bit faster, they grow to a little bit higher density, which ultimately helps reduce the cost because of reduced media usage. And so that's us. That's our company. And then we help companies rapidly implement these scalable manufacturing platforms so that they can focus most of their time on product development, less of their time on manufacturing process development, and then help them tech transfer into CGMP manufacturing environments. Because once you actually, for anyone that's actually gone through this, once you have a manufacturing process developed, you're still between 2 and 3 years away from treating a patient, because it takes a couple of years to actually establish those cell banks. It's a pretty big supply chain roadblock and bottleneck, and that we've again tried to remove. So we don't run clinical trials. The scale of manufacturing for our own products is in the single digit billions. That was one of your specific questions. But then we're helping our customers get to the tens of billions. And I would say that we haven't gotten that into clinical. Our customers have not gotten that into clinical trials yet. But we're hoping to get there, to get some of that running next year. And any of our customers that are in clinical trials today are in the cell stack type processes, more of the traditional processes where with lot sizes in the single digits.
Great. Thank you so much, John. I think we will come back to that scale question a little more in depth. Lisa, you are part of a smaller therapy company, and do you want to introduce what you guys do and what scale you guys are at right now?
Sure. I'm currently with Longeveron. We are a private clinical stage regenerative medicine cell therapy company. We were founded in 2015 for the purpose of developing and commercializing cellular therapy for aging-related diseases and conditions. Right now, we have 5 multi-center clinical trials ongoing in the U.S. testing our lead product in various indications, some of which are partnered with the National Institute on Aging. Our lead product is culture expanded allogeneic MSCs derived from the bone marrow of healthy young donors. And at this point, we have treated close to 300 subjects in various clinical trials. Early on, we decided to invest and develop our own product research and development and manufacturing capabilities in-house instead of outsourcing those. So we operate a CGMP compliant manufacturing facility in Miami, Florida. And that's where all of our product is made.
Great. Thank you, Lisa. And finally, John, from Mesoblast standpoint, you guys are probably, one of the more advanced, larger companies in this space. And what are your thoughts? Obviously, again, introduce Mesoblast for those who may not know about the space that well, but also talk a little bit about your general approach to manufacturing and kind of the scales that you guys are operating on.
Sure, thank you. Mesoblast is a mesenchymal lineage cell company, and we have two types of cells, two primary types of cells, the mesenchymal progenitor cells, which we have completed accrual of phase 3 studies in both chronic heart failure and in lower back pain. And we are getting ready to perform the second phase 3 study for both of those studies. In addition, we have the mesenchymal stromal cells, which you've heard about a number of times today by David, by Joanne, and the benefits and the activity of these cells against cytokine storms and effect in GVHD. These cells are obviously we have submitted a BLA, and we're in the middle of that BLA. FDUCIA date is coming up, and therefore have to watch what I say about that. But we're hopefully will be positive news, and we will be ready. We've been fortunate in terms of pediatric GVHD because it is a low requirement. There's not many patients, or I shouldn't say not many, but there's a low number of patients that require these cells. And so therefore the manufacturing capabilities, the scale up has been relatively easy to achieve. We believe that with the process that we've had, I will just emphasize that we've worked very hard on developing a very robust process that gives us the same results time after time. And I think we'll talk about that in additional questions. But we've got this robust process, which we believe if in one suite, we will be able to manage the requirements for GVHD in the US from that one suite that's in Singapore. So we've, in terms of what we've done, we've really looked at scale up and scale out. And initially we've dealt with scale out, which means we've developed a system where we can just keep building more rooms in order to handle the initial therapies. But we are also looking at scale out. John referred to the bioreactors, and that's probably the biggest one, is the scaling up is the bioreactors, which will give us many more cells. And that we're working on those too.
Great. Thank you so much. I want to kind of go to kind of keep on that scale theme for a second, and then we'll move to quality, for example. But say, again, John, maybe I start with you from the Mesoblast standpoint. You guys have trials going on in COVID, right? And that's
Yes.
That's already public knowledge. So there, that scale requirement suddenly goes up, and maybe even outside of the MSC therapies, in NK cells, in CAR T cells, when we go into larger adult populations, that scale requirements grow up. From your point of view, having been kind of a cell therapy industry academic veteran, how would you think the industry is dealing with the scale issue now? Are we on track to be able to get the cost down, deliver it to a lot more people? What are your thoughts in that scale?
So yes, you are correct. We've spent a lot of time over the last 3 to 4 months looking at scale up because of our COVID trial. And if the results continue as they have before, David presented 75% response in the COVID trial. We, again, will initially deal with it using scale out. And so we have developed the models, we've developed blueprints in order to be able to hit 10,000 patients in the first 6 months. It will require a lot of coordination. It will require a lot of money, to be very blunt, because we will have to build up things very quickly. It's going to require suites that already are available as opposed to suites that will be available in 6 months from now. And it's going to require a lot of investment or a lot of collaboration with the vendors of the raw materials. And I see that as being the most difficult one at this point. The raw materials right now are going specifically to COVID, but in order to hit 10,000, I think we're going to need a lot more and a lot more collaboration with the vendors. So I would say those were the 3 points.
That's great. And maybe others like, John, your company certainly, John Rowley, your company certainly thinks about this scale issue a lot, as you mentioned. But as you think of, diseases like COVID maybe, or arthritis, or where there is a lot larger patient population, and we gradually come out of the pediatric indications, where do investments need to be made in terms of scale per se? John, you are muted.
Okay, you're asking this, John, right?
Yeah.
Yeah.
Okay. So when I think about lot size, and these patient populations where you could be treating hundreds of thousands of patients a year, right? If you back calculate, let's say you want to treat 100,000 patients, or even 50,000 patients a year, you need a lot size that you can imagine manufacturing to that scale, right? And so it's all about dose. It's all about lot size. And we actually have written some blogs on this. If you go to the RoosterBio website and do our blogs, we actually have blogs on specifically coaching people through this, depending on what your indication is. But let's say you have 50,000 patients, and let's say your average dose is 100 million cells per dose, and that's probably on the low end of moderate, right? If you look at all the clinical trials, the average dose is 300 to 400 million cells per dose, but that's over lots of different indications. And so 100 million cells, and for 50,000 patients, right? You're not going to want to do more than one manufacturing lot a week, right? So even if you do one manufacturing lot every 2 weeks, that's 50,000 doses over 26 manufacturing lots, 25 manufacturing lots to make the math easy, right? And so you need at least 2,000 product doses per lot, right? And so 2,000 product doses per lot, and I should have done this math before talking about it, because now I have to do math in my head. But 200,000, 2,000 doses per lot times 100 million is, I think, 200 billion cell lot sizes. Right. And every dose is usually at least 30%, if not 50% overfilled. So add 50% on top of that. Manufacturing, if you just manufacture 200 billion cells, and you're going to lose 20 to 30% in your downstream processing. And so you're only going to be vialing 150 million, right? And so there's all these things that to account for it, to really get to your lot size. And again, I've got this written out as education in an educational blog series on our blog for anybody to check out and kind of go through the math yourself. But you're looking at a commercially relevant lot size for a real indication to be on the order of 250 to 350 billion cells. And when today's lot sizes in 10 layers are, between 10 and 15 billion, to get to a 50 liter bioreactor, right? Which we've been in, with the best yield that we got there, we got a little over 30 billion cells out. Then you're still one log below this, right? So you got to get to this 500 liter scale, which is when anybody thinks about the different unit operations to do a 500 liter bioreactor. I mean, who knows how to manage, 300 to 500 liters of of harvested MSCs and quenched trypsin or triple E.
So you would think that what John was mentioning, really a combination of scale out and scale up is sort of the short-term strategy. You can have 10 such bioreactors if the investment is there. And there needs to be some manufacturing investment in the field for us to get to 50,000, 100,000 patients. But certainly 10,000 patients is more doable at the current stage in a 6 month to a year manufacturing head start.
I mean, 10,000 patients. Again, it all depends on dose. 10,000 patients from a 10-layer process is you're churning out several hundred product lots in a year, and it can be done, right? I've been in an operation that was planning to do it 10 years ago, and and so it is possible. It's a big operation. Bioreactors are definitely the final answer, but I am 100% confident that the field can do it with today's manufacturing technology.
Yes. That's fantastic to know. Maybe I can go to Lisa next and really kind of switch the topic a little bit, Lisa. From your point of view, running a smaller business, what do you see as the challenges in the CMC space that needs to be solved for, say, treating 10,000 patients or really getting through the regulatory approvals and so on? How do you guys think about the challenges?
Well, as far as making a product that's consistent through all lots, you have to have set release criteria. You have to have set specifications for your raw materials and any disposables that you're going to use. So you have to have your critical quality attributes in place. Everything has to be set appropriately. You can't just make changes on the fly because something doesn't fit into what you've already got set up. You have to work within certain parameters, and you rely on your quality group to do that. So quality obviously provides a check on manufacturing to make sure that each lot is consistent. And in order to have consistent lots in a GMP facility, you have to have highly trained staff. You have to have consistency of process, and you basically have to do your best to do the same thing every time in order to produce a product that's very similar that will meet your release criteria. Otherwise, you're not going to get very far.
Right. No, thank you for that. In the noon session with Peter Marks and others, we heard a lot about sort of the manufacturing process variation, creating different cell properties and so on. I'm sure, obviously, as companies, you guys think a lot about quality, and that is certainly a hallmark of the CMC aspect of cell therapies. How do you see, maybe John McMannis can comment on that, this whole idea of identification of critical quality attributes and critical process parameters, and eventually understanding mechanisms of actions. How you as a company and the manufacturing units work in tandem with the clinical side, maybe, to really address those CQA, MOA type questions that obviously very important from the regulatory standpoint and the product standpoint.
Sure, they are very, very important. And the consistency of a product, as Lisa mentioned, the consistency from batch to batch is extremely critical. The other difficulty with a cell like the mesenchymal stem cell, as you saw this morning, I believe it was Josh who presented the mesenchymal stem cell, and then all the different factors that can be secreted of it, and then the activities of that cell, depending on which way it wants to go, whether it works with macrophages or T cells, et cetera. And so you could, when you're looking at mode of action, block one, but not block the other and still have the effect. And so with cellular therapy, definition of the mode of action is extremely critical, but it's extremely difficult to define. And we have worked on ours very closely in discussions, et cetera. And so it's just a hard thing to do, but it's a critical because you need, again, as you produce product over years, you have to make sure that there isn't any drift.
No, thank you for that comment. And I totally agree with you that really the field is just beginning to understand the complexity of trying to figure these things out. And when you have a product like a cell therapy, which has likely multimodal mechanisms of actions, and you're really looking at a multivariate space, then the analytics of that needs to change, the data analysis part of needs to change, and so I think we as a field are still understanding a lot of that. Maybe John Rowley, when you guys go from, say, the smaller scale to the larger scale to the bioreactors, you guys, what are you looking at in terms of consistency and comparability? Product comparability, which is obviously when you change a process parameter or a scale, you have to go back and think about product comparability. So how do you guys think about that in terms of product comparability?
So great question, right? Obviously, all the safety stuff has got to be there, right? I mean, you're definitely looking at the yields and everything. So you need, good single cell suspensions. You need high viability out of the cells, and so all that is, I call that table stakes, right? Now, since we're not a therapeutic company, we don't have our own potency assays. And so what we've used when we've scaled up to 50 liters and compared that to 10-layer culture, we use many of the general MSC fundamental markers that people do look at for for some of the mechanisms of action, such as immunomodulation, right? So we used IDO as a measure, inducible IDO. When you tickle the cells with interferon gamma or TNF alpha, they start expressing IDO, and so we've shown from a flask to a 10-layer to a fed batch bioreactor, the inducible IDO is very comparable. We also looked at the half a dozen angiogenic cytokines on a picogram per cell per day level at the of the cells that have been harvested and again froze down, treated like a product, right? You vialed it up exactly the same way. Thawed out, do your CQAs on things like angiogenic cytokine, as well as the multi-lineage differentiation. So we've done all of that. I would say that is what we've done. Until you actually have a very specific indication, it's hard. I'm sure Longeveron and Mesoblast have very specific potency assays to do it with. I would like to add just one thing on the CMC challenges of specifically for MSCs, is that when you're on the market and trying to treat tens of thousands of patients, you have to be managing several donors per year, right? Now there are I think the IPS MSC will eventually solve that. There may be one donor. Managing multiple donors, even for 50 to 100,000 patients, you're really talking 5 to 10 donors a year. It's not like it's hundreds and thousands, right? So it is manageable, but you do need to be able to identify not just the good donors, but also what a bad donor is. And you never want to get that bad donor into manufacturing, because once something's in GMP manufacturing, it gets really expensive. And so just having that strategy for understanding what donors are going to be good specifically for your potency assays is definitely important, because I like to say the best donor for cartilage regeneration is not necessarily going to be the best donor for GVHD, and there is a lot of donor to donor variability, and I think that is a CMC challenge to manage during clinical development as well as then as you go to market.
Great, thank you. And Lisa, coming back to you. You guys are looking at certain therapeutic indications, so you're developing your potency assays and your release criteria and so on. And is that, I think you mentioned this before, but maybe you can elaborate a little bit, sort of, again, in a broad stroke rather, as to the strategy that you take for comparability when you're maybe thinking of scaling or changing process and so on and so forth. How do you think about making sure that the product is comparable or remains the same?
So you're asking if how we determine if a product is comparable lot to lot?
Correct.
Yeah.
Or as you think of scaling.
Yeah, that can be difficult. I'll go back to John when he referenced donor to donor variability, because a lot of times you'll have a donor in culture that looks like it's going to be a great producer, and maybe it does produce a lot of doses, but after being cryopreserved, it maybe does not thaw well. So then you've got a situation of what looked like a great donor on the front end is not so good when it comes to thawing and formulating your product as far as viability goes, and maybe some other release tests. We haven't had much of a problem in that respect. You're always going to have some level of failure with donors because each person's different. But until you know specifically what you're looking for, and you can test your donor for the specific markers or whatever you've come up with in your research to treat the indication that you're targeting, it's going to be difficult to always weed those out.
Yeah. No, thank you for that comment. And obviously, we have been mostly talking about allogenic products and MSCs in particular, but there are plenty of autologous cell therapy products, even CAR T cells, for example, that where the variability becomes much more challenging, even much more challenging than the 5 to 10 donors that John was talking about, and really thinking about donor characteristics and their relationship with the product characteristics within a controlled manufacturing process is something we need to invest as a field, and we don't know a lot about how a priori can we predict what donor is going to yield what quality of product. I think those are very, very interesting aspects of cell therapy that we need to, as a field, figure out going forward. I wanted to come back and talk to you a little bit about your kind of viewpoint about sort of public-private partnership in this space, right? I mean, it's a complex product. For companies, it's a high-risk product. On the other hand, there are a lot of promise, not just on T cells, immune cells, NK cells, MSCs and others, but a lot of activities in the iPSC space right now, which are very promising. Where do you see the role of federal partnership with industry and maybe industry academic partnership? We heard from the FDA today, we heard from congressional staffers today. There is really a nexus, of course, where healthcare can be moving forward faster through partnership. And I know Peter Marks and the FDA commissioners talked a lot about collaborations, cooperations, and partnership, even bringing the FDA early on in that process. So I want to go to each of you in sort of series here to get your kind of broad thought about federal partnerships with industry and academia and academic industry partnerships and so on. Maybe John McMannis from Mesoblast, you want to comment on that first?
Sure. Obviously, it has been brought up a number of times today, and I agree. We need to have it. I almost think that CIRM is probably a great example where it was a partnership between, yes, the state government and the research institutes, but they initially put a lot of money into building the structures so that the trials could be done. And then I think today you heard some of the benefits and some of the rewards from having done that. We aren't there. Other than at the state level, I don't think we're there. I don't think the collaboration is there. And and I think it needs to be worked on. And I do like the suggestion that the agency could be a facilitator. Andy was the one that suggested that they facilitate the working among groups. Obviously, they did it with the plasma studies, and I think they could start doing it with some of the cellular studies, and getting to go. The other thing I would bring up is I was excited to hear that NIST is getting involved in this, because I do believe we're going to need control and reference standards for the various things, and to have NIST in there getting standards ready that we will be able to compare. Even if we don't collaborate, at least compare one trial to another. But if we can collaborate and then show, yes, the products are both the same, that will be even more powerful.
John Rowley, you want to chime in a little bit? And I know you as a company have worked with federal fundings. You mentioned MTech. You work with academics quite a bit as well. So what are your thoughts on partnerships? John, you're muted.
Sorry, I think someone else muted me.
Go ahead.
I didn't mute myself. So it's overall good, right? I mean, be able to fund some things that are some pre-competitive technologies, right? That all companies can use. I think one of the things, and we've been, part of some things like the CMAT, the Cell Manufacturing Technology Consortium, and some of the things that we've brainstormed, there, even, pieces of the supply chain, right? Things like GMP growth factors are so challenging. There's a bunch of vendors, and you don't really know what the FDA is going to say about any one of them until you actually use it, incorporate it into your product, and then present it to the FDA. And there's 10 different reasons why they can say, oh, this is no good. And if you've already made product with it, you're at a tremendous disadvantage, right? And so I think we do need some transparency in some of these challenging things, but the FDA can't choose one company's product over another, right? So there are a lot of challenges here, but I think there's a lot of opportunity in just the whole supply chain of regenerative medicine needs to get industrialized, for our next generation therapies as we go commercial. Unfortunately, I think we're just stumbling through it, and I think that these public-private partnerships, could be hitting on these things. Now they are funding things like these like the 50-liter bioreactor project for us. I mean, that was tremendously helpful. That came with MTech funding in collaboration with Institute for Surgical Research, which is part of the U.S. Army. A nonprofit, GenCure, who's a contract manufacturer in this field, as well as another small company, Stembiosis, and so they are allowing for work that does cost several $1 million. That's the challenging with developing bioreactors is it's millions of dollars in development, because 50 liters of media is not cheap. Even for a company that makes media, we couldn't afford to run our own 50-liter bioreactor, and so getting this funded by government was really one of the first ways to get a generalized process available. There's trying to apply that to things like T cells and dendritic cells and then iPS cells. It does get challenging because each cell is unique, has its own challenges, and what you learn from MSCs isn't necessarily applicable to iPSCs, and definitely is not applicable to autologous T cells. And then even with MSCs, while it was a several $1 million program, and we did get this a 50-liter process, as I said, a true commercial scale process is 500 liters. It's 10 times. That is 10 times the cost of a 50-liter process, right? And so the cost of these things does get exorbitant. And I think just like in the biotherapeutic field, the monoclonal antibody field, I really don't think you're going to see true process innovation happening really rapidly until we get the first blockbuster drugs. And we need Mesoblast or Longeveron out there making $1 billion, and that allows them to spend tens of millions on their own manufacturing processes. So we're still in the early stages of a field. I mean, I've been in this field 20 years, and it kills me to say we're still in the early stages, but just commercially, we need some significant successes so that there is money then to invest in these bioreactors.
Yeah, no, I really, agree with you on the fact that obviously we need investment, we need public-private partnerships, and we need to really base everything on the fundamental science, but really collaborate and work between industry, academia, and the government. John, thank you for bringing up NIST and the role of standards in all of this. Because that is huge as we really think about collaborations and standardizing methods and analytical practices. I want to quickly check and remind the audience about opportunity to ask questions to the panelists. I don't think I see any questions for this session right now, but those of you who are watching live, there is a chat button next to you on the right side, but also in slido.com, you can use the event code T as in Tom 992. So that's hashtag T992 to submit your questions, and I'll try to get as many questions as I can as they come in. Maybe in the time that we have remaining, I can go back to Lisa a little bit. And ask you about, you guys have collaborated with academia, I believe so. And what is your experience in bringing cell therapy from academic labs or clinical centers into a product for a company? Can you comment on that?
Yes, I can comment on that. Sometimes that can be difficult depending on the level of the academic institution that you're working with, because there's different ways that people work, and academia is not like a GMP facility. So a lot has to go in to ensure that you're using components that are safe and they have the required release criteria in order to move those into a GMP facility and make them releasable. And the processes, and you have to listen very carefully, because a lot of times in an academic setting, researchers, they'll change on a dime, and you can't do that when you get into a GMP facility, because then you're not going to make consistent product, because you're constantly changing up your process. So that makes it difficult. So luckily, all the interactions I've had with research institutions have been pretty good. They're good to work with you. They know that it's different than what they've been doing on their bench. So it can be a little challenging, especially if you run across
So what's your advice to academic and phase 1 early stage clinical researchers who's obviously, all of our ultimate goal is to get these products to patients, right? And get it to a real product. So what would be your advice to trainees and kind of the emerging workforce and the academic folks to think about early on as to make that process easier?
Well, early on, choose the right components. Don't use something just because it's what you have in your refrigerator or your freezer. Make sure that you write down exactly what you're doing so you can transfer that. Because if your ultimate goal is to get into research and phase 1, phase 2 clinical trials, you need that information to pass on to your CMO or whoever you happen to be collaborating with. Don't shortcut. Don't realize that it's different than what you're doing, and be open-minded about it. So other than that, it's just open-mindedness, really.
Great. Great. And maybe John McMannis, you and Mesoblast has a strong and long history of academic collaborations. And what are your thoughts on accelerating products to market? Now that we have a lot more tools, we know a lot about cells and cell therapy. We are still obviously don't know a lot as well. But still, what do you think can accelerate that timeline to a product?
To a product. So I'm in a very unusual situation in that I was doing a clinical trial at an academic center with Mesoblast and it was a phase 3 study, by the way, and we were doing it and then moved to the other side and suddenly realized all the requirements the industry partner was going to have to do, and went back to the academic side and said, okay, we need to work on this more. So, but to accelerate things, I think that we need to get, I guess I'll take the same approach that the FDA used this afternoon in saying that really collaboration and discussion needs to occur early. So if they go all the way through the product design, protocol approval, and then come to the industry partner and say, here, we want you to use your cells to do this study, and we want you to look at these things without any discussion, then I think it will be slower, because I think the industry partner will probably have more knowledge of what else has occurred already with those cells and be able to say, well, this may not be the best marker to look at, but these would be the better markers, or this is probably a potency assay to look at. So I think it's collaboration early on. I think it is a willingness of both sides to work well together. I think you do see academic centers these days, their IP group always thinks that they have a lot more stake in it, and sometimes they do and sometimes they don't, and so it's got to be a collaboration between the two, and I think that will slow things down, speed things up if the IP can work faster.
Thank you so much. And I think, on that note, there was a similar question in the chat room here is, aside from the funding issue, investor-related pressures and IP often preclude this multiple collaborations.
Mm-hmm.
And I think you addressed that quite a bit. There are a couple questions for John Rowley on the chat, and I'm going to throw them to you.
Sure.
We have about 3 minutes left, so maybe you can take a minute to answer them. One was, how far away are the bioreactors? And the related question to that is, what number of population number doubling is acceptable before loss of potency morphology? And how has this changed with xeno-free media compared to FPS? So some technical questions.
Okay, so bioreactors, how far away are they? I think that there'll be clinical doses manufactured next year in bioreactors, and that might, be more extracellular vesicle products that are derived from MSCs, as opposed to MSCs, the straight MSCs. But I think that the MSC ones are not, are not far. None that I know of are actually tech transferred into CGMP manufacturing at this stage.
And then the second question was on the PDL.
PDL.
How much PDL you can go to?
PDL is, it depends. Mesoblast and Athersys have very different, I think philosophies on PDLs. And it really depends on the product, on the process. Cord MSCs go to higher PDLs because they have more PDLs available, than the adult bone marrow. So it really depends. I would say for your standard bone marrow, most things that I have seen keep total population doublings, post mononuclear cell isolation at around 20, right?
Okay,
Matt,
I'm going to have
yeah.
to stop because we are out of time right now. So John, thank you so much, and John McMannis and Lisa McClain-Moss, thank you so much. This was a fun conversation. We have a lot more to learn as a field and a lot more to do as a field, of course, and I encourage everyone to think manufacturing, think quality, think scale early on for the cell therapy space. And with that, I wanted to thank the organizers for giving us the opportunity for this discussion. And thank you, everybody. Bye.
Thank you, Chris, for leading it.
Thanks, Krish.